Neurons are unlike virtually all other cell types in the body. They collect and transmit information in the form of electrical signals, which travel along the neuron's membrane. Electrical signals are then transmitted from neuron to neuron via specialized cellular structures known as synapses, which use neuron-specific chemicals, called neurotransmitters, to transfer information from one neuron to the next. In the next several videos, we'll take a deep dive into the cellular and molecular bases of the action potential generation and synaptic communication, how channels or pores in the neuronal cell membrane create action potentials, how the action potential propagates in the cell body along the neuronal axon, and finally, how action potentials are transmitted across physical gaps called synaptic clefts between one neuron and the next. Understanding action potentials and synaptic communication at a molecular level is important because there are many different types of neurons, and molecular level processes ultimately determine the functional properties of each neuron and its ability to contribute to neural computation. Also, many common toxins that you or your students may be familiar with will operate on or interfere with the molecules responsible for initiating or propagating action potentials and interneuronal synaptic communication. Similarly, some neurodegenerative diseases result from a failure in the propagation of action potentials or synaptic transmission. And hopefully you'll find these molecular details interesting in their own right. But before we get into those details, I'll start with a big picture overview of how neurons communicate with one another. This overview is the key to understanding how the brain processes information, and you don't need to fully understand all of the underlying molecular mechanisms in order to appreciate the overall concept of information transmission between neurons. First of all, information flow through neurons is unidirectional. Neurons gather input or information at one end and send that information to the other end, which transmits it to the next neuron. Neurons have specialized anatomical features that facilitate each of these stages of information transmission. Information is collected in dendrites, sent down the axon, and then transmitted to the next neuron at the synapse. Note that axons can be relatively short, in which case the next neuron may only be a few hundred microns away, or very long, say in the case of a motor neuron sending information the entire length of the spinal cord, or anything in between. 2. Neurons combine many inputs to generate a single output. The exact number of inputs and outputs can vary widely for different neurons, but a neuron can easily receive thousands of inputs and send its one output signal to thousands of other neurons. 3. Connectivity shapes a neuron's response properties. Neuronal connectivity, where a neuron gets its input from and where it sends its output, is not arbitrary, but is shaped by a complex interaction between genetics and experience. Connectivity patterns give rise to many of the unique properties of different neurons and determine how they relate to the sensory world or how they drive behavior. Here you can see just a few neurons of different types and how they connect to one another in very specific ways, which in turn will shape the neural computations that they perform. Clusters of interconnected neurons are often referred to as forming a neural circuit, analogous to electrical circuits formed by transistors or resistors that make up modern computers. So to summarize those three key points, information flow in neurons is unidirectional, neurons combine many inputs to generate a single output, and connectivity between neurons is what will shape the neuron's response properties. That's the big picture that I want you to keep in mind as we dive into the details of action potentials and synaptic communication.